So this is the next big things in cytometry interview webinar series hosted by the International Society for the Advancement of Cytometry, CYTO Innovation Committee. Today we have a very interesting topic. We're joined by Kevin O'Holleran, the CEO and co-founder of ZOMP, and we're interested to talk about 3D imaging cytometry. Now, as everybody has seen, imaging cytometry has been a big trend in the last few years. All the major vendors are racing to adopt 3D imaging and 3D spectral imaging, so far all the major technology is two-dimensional, and as everybody knows, cells are three-dimensional, and it's a big curiosity whether it's possible to go into the third dimension. And now Kevin is I'll hand over to Kevin in a moment. Kevin and ZOMP are doing it seriously, and we'll hear about their efforts and why they think it's the next big thing. Now, we're open to question and answers. This will be a conversation, so please feel free to ask your questions throughout, and we will read them out and weave them into the conversation. Please use the Q&A box, not the chat, because otherwise your question may be missed. Now Kevin will give a presentation, but not at the beginning. It'll be about halfway through, so hold on for that. Well, I'm Samson Rogers, and my co-host is Catherine.
Catherine, hi.
Hello. And so first, and then our guest is Kevin. So over to you first, Kevin. Could you introduce yourselves? Introduce yourself and how you got into 3D imaging flow cytometry.
Well, first off, thank you for inviting me to be here today. It's not the kind of event I've done very much of. We've spent 2 years kind of buried in a basement trying to get things up and running and working, and out there into the world in local labs. So it's really great to start, contacting the community more and actually be a bit more visible and talk a little bit about the exciting technologies in the space that we work within. I'll try to keep it the brief version of how I arrived here. Originally a laser optical physicist in the University of Glasgow who was initially fascinated by tying knots in light, which won't make much sense to anybody, quite an esoteric field of study, kind of fundamental physics of light. And that simply wasn't enough. It didn't have enough impact. So I finished my PhD and joined a startup doing mathematical modeling, very early kind of machine learning AI back in the 2000s. And that still wasn't enough for me. It wasn't enough just to write programming and do maths. I wanted to do something, have more impact on something very practical. So I saw a role being offered at the National Physical Laboratory in 2011, and that was about the era that super resolution, really high resolution 3D imaging was really booming. All these techniques had suddenly exploded into the field. And there was a real need for physicists and engineers who could understand that world, but also were interested in wanting to apply it into biology. So I joined the National Physical Laboratory to develop some of the UK's first super resolution microscopes. So these are microscopes that either determine molecules to within 10, 20 nanometers in a single cell using fluorescence, or 100, 200 nanometers over a larger area of maybe a few tens of cells, but they're really focused on that kind of small number, very, very high resolution. And that was sort of the gateway for me into biology, into microscopy. And I very quickly moved on to the University of Cambridge, again, to get even more practical, because the National Physical Laboratory was a great place to hone metrology, sort of define how these microscopes work, how they measure things, how do we make that traceable? It's a new technique, so how do we get the protocols right? But it still lacked the kind of application in the trenches application interface I was looking for. Uh, so the University of Cambridge was creating a new lab called the Cambridge Advanced Imaging Centre. That remit was not to just make microscopes available and kind of charge for usage to biologists, but to develop new microscopes to answer the most painful questions that some of these researchers have, but find commercial instruments limiting. So we first started on super resolution methods, light sheet methods, and these are microscopy methods which push the boundaries on either resolution or speed in some fashion. So that was the bulk of 10 years of running a lab there and doing research in the space of cells and tissue, model organisms, zebrafish, Drosophila, organoids, kind of synthetic or like 3D models of real tissue. And even then, I still had this thirst to have. This is still not really having, I feel like having impact. This is, I'm working with some of the best biologists in the world, but you're working with two or three people and it culminates in a paper and writing another grant. And it still felt like, there's more than just this. And we took one of our methods we were working on, which was light field, single molecule light field microscopy, which was a method that I designed to try to speed up these really high resolution methods. And then we thought, what if we turn the dial down on the resolution and just really go for speed? Uh, and that's where we started looking at cytometry and thinking, well, if we kind of aim at micrometer resolution and stop worrying about 10 nanometers, we can do this pretty quickly. We can maybe even do it in flow and we can do hundreds or thousands of cells. So then a very long story short is talking to the tech transfer office, raising capital from Zeiss Ventures, University of Cambridge, and some London venture capital funds, Foresight and BGF. We founded the company and spun it out, and I spun myself out with the company. I didn't think that staying as a professor and trying to run a company was a viable path to a successful company or even a healthy life. So I left to focus on the technology, and here I really found that match for what I was looking for, which is taking a really bleeding edge technology and trying to match that to what a much larger community of people are saying they need in their sciences or need in their area of work. And so I really believe that this is we're heading in the right direction, and that this type of technology is not just necessary but inevitable, and that's where we are today.
Yeah, great. Yeah, I was curious. What did your users at the imaging center make of that when you started telling them that you wanted to turn down the dial on the resolution and increase on the speed and look at populations? Were they into that, or was that for somebody else?
No, that's music to their ears.
Mm.
All too often, engineers and physicists in isolated labs, um, for example, like the National Physical Laboratory, great, amazing, world-class facility, but it's pretty thin on the ground in terms of biologists or life science applications there. And if you're not having that day-to-day contact, and the mindset is very much one of engineering and physics, there's a focus on things like resolution and milliseconds and nanometers. But you could take someone that's a leader in their field in zebrafish biology, and I asked them, when you're setting up an experiment, so just so I get this right, how big is the zebrafish eye? And they're like, oh, I have no idea. Like, it's not it's not a space that really solves anything for them. You really need to ask in terms of the genetics they're studying or the physiology or the phenotype of the disease. There's a diminishing returns to chasing resolution nanometers. And so talking to lots of labs and listening to what they really want, we found that they wanted the ability, this was in my lab at the time, to not get high resolution, but to perturb samples. Like, I don't want to just see it. I want to activate an optogenetic module, or I want to use a laser to cut something, or I want to, well, they wouldn't know the laser. They would say, I want to damage this tissue. How could I do that? And we as the
Sure.
engineers would figure it out. But resolution was never really top of mind for them, but it might often be the kind of tool or solution to the problem. So it's sometimes not useful just to keep chasing that metric because it's one that's won Nobel Prizes or is a very easy proxy for progress.
Sure, yeah. So very, very clear and a good segue into cytometry. So, I mean, our first question is, um, well, what is 3D imaging cytometry, and how does it differ from what say the widespread imaging tech imaging flow cytometry techniques that currently are nowadays?
Well, I could, I guess, say it does what it says on the tin, but it 3D imaging cytometry is just one subset of what I view as the larger field of spatial cytometry. And standard cytometry is spatial cytometry. You talk in terms of pulses and areas and heights, but that's simply a one-dimensional image. You sample a structure along its one direction, and you've lost all the information in the other two directions. So it's not very easy to think of it as an image, but it is exactly a one-dimensional image. So that pulse that's often used to gate out doublets or things you don't want to see, or it can even be used maybe cleverly to see if you're actually looking at the membrane or something internal, looking at the shape of the pulses relative to each other. That is spatial. So my view of this is that this field has always been spatial to some extent, just in a very light way, because what's more prioritized than spatial data is speed, right? You've got enormous populations of cells. You need to get through thousands, millions, potentially more if you're going through a clinical trial in many patients. It's just not viable, to go through collecting, or possible in the past, to collect all that image data. So, passing cells or aggregates or other things quickly through a laser, you get that pulse, and that's a one-dimensional image. So your question of to what is three-dimensional cytometry, I just view as the inevitable end point of having better technologies, better hardware, better dyes, and better label-free approaches that, given the availability of that data, it's going to be enormously useful. And it's not simple, but it's been a case of barriers to make that possible. In fact, one of the original imaging cytometry papers is in the early 70s. It goes that far back. And if you look at that paper, you'll see it's a three-dimensional cytometer that they actually construct two images from orthogonal axes. So kind of the minimal needed to call it 3D. And no one's really in that context thinking of 3D. They're just thinking of the cell has an entire shape, and I want to know, is it elongated or is it actually two cells or not? So I better take it from two directions. So it's from early on as the 70s, people have been trying to get this kind of data, and I would say it has been hardware, software, lasers, detectors. There's a long list of things that just weren't at the right quality or capability to achieve what we can do today.
Yeah.
And with
Well,
what you
very,
can
very.
do today with your new technology, what do you think are the biggest questions that can now be answered because of it?
That is a tough one, and one we have to face a lot with investors as we continue to grow the company, because there are so many answers. It's an area where the applications are, as we'd say in the UK, a mile wide but just an inch deep. If you were to ask me what's the key applications for a microscope, it's such a well-used technology that's been watered down to what might come across as quite a pithy comment to say that it's just to look at things and see where they are. So you're getting to a point where we want to transition from saying we just have the presence of a given receptor or the amount to the location, the where. Now, what applications is that where useful? So you get into applications that you'll have seen that are well trodden from people using the image stream and now the FACS like translocation assays, anything looking at trafficking, anything looking at particularly live changes in cells. You might go into areas that then are not quite classically cytometry, and that people interested in organelle health or shape, they want to see how that changes over time. And they've been limited to, this is on the other side of the spectrum, they have used microscopes inside out for decades, and they've been limited by the statistical power and having to look at adherent cells, on glass slides. So the applications, in my view, depend on the interface that you're looking at. Are you looking at people that traditionally use microscopy tools and have been limited by statistics, or people that use cytometry tools and they've been limited by that location question, such as is it really on the surface or not? It could be quite basic. I don't really want to know within 100 nanometers where it is. I just want to know, is it external membrane bound or has it been internalized? Just a very simple question like that is still a location-based question.
Could you give us a flavor of the why now? I mean, as an addition to that question, because I mean, of course, I mean, you mentioned a lot of these. I mean, these biological measurements are, like co-location and localization to organelles and changes and so on. I mean, actually, in conversation with you, you mentioned also that there's a synergy here between. I mean, it's not just seeing a lot of images, but also the computational, the hardware, and the analysis techniques that are actually all necessary. Could you, yeah? So the why now? How does this all come together?
Oh, you could take your pick. I mean, to go to the more boring ones, you might not expect it, but I'd say hard drives, right? The performance and speed improvement in how fast you can save gigabytes of image data to hard drives is something that did enable a whole suite of microscopy methods about 10 years ago as well. These scientific CMOS chips that have been increasing in quantum efficiency. I'd say, I have to maybe remind myself my age, because I would say 5. It's probably more like 10 years ago, 15 years ago, that these cameras were creeping up from 50% quantum efficiency to 70, and everyone's buying up these cameras like mad to put on their microscopes. And then within a space of a year, there's a 90% and another 95%. So now it's just standard if you're buying a really high-end microscope that you're going to put a 95% quantum efficiency detector. And by that, I mean 95% of the light that's actually being emitted by your fluorophore or collected by your optics is detected and used as a signal. So that is something that traditionally stopped cameras being used over photomultiplier tubes or photodiodes, is that splitting the light up into your pixels, and then those pixels only collecting 30 or 50% was just such a painful price to pay that you wouldn't really see very much. And that's what has changed dramatically on the sensor front to enable direct imaging cytometry, where we collect on a camera sensor the image of the cell. Like I said, the hard drives need to be fast enough then to dump that data for you to either analyze. Even when you had that data, I'd say up until even 3-4 years ago, analyzing that was tough, right? You'd have to have very bespoke code. You'd have to have your own machine learning, image-based models, your own software. But the rate at which machine learning has boomed, and particularly automated kind of image analysis, a lot of that barriers we found it's just fallen, it's just crashed away. And it's now become quite straightforward to set up very bespoke pipelines on any kind of bespoke image analysis you might ask of us, and to return very quick analysis. Again, with the modern day GPUs, so the boom in AI has pushed GPU performance, through the roof. So all these peripheral fields like imaging and microscopy are benefiting from all these things that are happening in the computational space and the software space. Then in tandem to that, you have the improvements that you will probably know, and everybody watching will know far, far greater than I, are the improvements that have been happening in the palette of dyes and the different optical tools and chemical tools that you can use, and the whole spectral palette and how you can choose where you label and what, and where do you position it on the spectrum. That all those things have come together. Oh God, the list goes on. Lasers, one. If you're buying a confocal laser 10 years ago, it used to come with an enormous box next to it with all these very sensitive lasers that were very expensive. Diode lasers now are amazing quality and relatively cheap. You get very good power on them. So, in our prototype down in the basement here, we just stuck 5 lasers on that as a starting point because it was quite simple to get the wavelengths from UV right up to far red and the power we needed. So I'll stop there, but you can see it's not that the technique itself is maybe new, although we have some, very innovative optics, I would say. The seeds of those optical ideas are still quite old, and it's only really been made possible. And I think companies like Zeiss invested in us because they see it's very timely, and you see that in, other companies, whether it's like DeepCell, ThinkSight, the BD or the water systems now. That this is all kind of happening at the same time because of all these technological advancements in adjacent fields.
Yeah. Well, I think, I mean, highlighting one aspect that you talked about, the data analysis part of this. I mean, this. Of course, this is experiencing I say data analysis because of spectral, especially because of spectral flow cytometry is experiencing an explosion of cytometrists getting into all the techniques of dimensionality reduction and clustering and so on. And there are workshops at CYTO about how to do it properly and so on. So this is all rapidly developing, and I think, as you said some time ago, this is very relevant for imaging cytometry as well. And I think you speculated that there is say adding that dimension, adding more imaging information would actually compete with the need for spectral information. So say the leading vendors at the moment, they've taken the approach that everybody wants, full spectrum plus imaging, but say limited imaging. But perhaps you see the balance the other way. More imaging would yield a great.
I will use the right word of balance there. I'm not going to be arrogant enough to say, the imaging is going to replace all that. That would be complete nonsense. But there is an element of like asking why. Sorry, I refer back to when, if I ask someone on a microscope, what resolution do you need? It's the wrong question to ask. How many colors do you need? You should be asking, like, what do you want to discriminate? And if you're trying to discriminate cells that have a different diameter or have a different nuclear shape or have some other morphological difference, there's no need to spectrally label it differently. You could use the same color. I'm not saying you don't label it. You've just got extra dimensions of information where things separate naturally. Now, in cases like, in immunology, if you're looking at a T and B cell, maybe you can't do that because they're too similar. So there are cases where you will absolutely have to use, say, a receptor to distinguish a certain type. But there are cases where, say, like a viability dye, that simply the shape and the morphology of the cell will tell you about its viability, potentially even just labeling the nucleus. So leaning into some of the things that people have done in the cell painting world, by labeling organelles, you can infer quite a lot about the state of the cell. So you couple that with, say, stick with immunology, stick with these CD markers, couple them together, you just create a whole new playground you can work in. Like, what panel should I build? It's not just about thinking about these markers, it's thinking about the morphological space which the cells live in. So if you imagine the full lineage tree of, say, white blood cells, you could look at that and say, which subsets of these just look very different. And that could change the way that you think about labeling it. So you may go down from, 20 colors to 12. I'm not saying it disappears. And then there's another aspect to this, which is that's just using a human understandable aspect where I can say its size, it's something very clear. Once you have thousands and thousands of spatial parameters, we haven't mentioned this yet, once you have spatial, it's not just a case of I've added a second dimension and a third dimension. Because I've added all these pixels or voxels, the parameters that I can extract becomes near infinite. So it goes away from, if I have a pulse, there's only so many things I can say. I can say how tall it is, how wide it is, how wobbly it is, how noisy it is. And something that mathematicians call the curse of dimensionality. Once you add dimensions, the number of measurements you have rises exponentially. And the number of combinations of those pixels and operations you can apply to them rises even higher. So you open
That's what
yourself
you
up
mean,
in this
yes.
space.
Yeah, so I mean, clearly there's a great, potential amount of data there, of course, yes, yeah. Actually, so just to come back to one point you said just now, have you thought of doing that as a demonstration, say what immunophenotyping can you do the lineage of white blood cells to show that there's rich information there that say competes with people's 50 or 60 color immunophenotyping
Mhm.
panels.
Absolutely. Now, tackling that's not sort of top of our priority list. What we haven't covered yet is the scale of ZOMP. We're 5 people,
All
five
right.
people, and
Yeah.
2 prototypes in the basement, and we're still prioritizing getting a lot of the straightforward engineering, to make a turnkey instrument working. But on that horizon of 5 years from now, I see this not as ZOMP on its own, but the imaging and spatial cytometry field is going to progress where people begin to understand how to build their panels now that they have all this new information. And we did find in some early experiments that simply using in cell painting, they create something like 500 parameters. There are various measurements like eccentricity, areas, granularity. They very quickly become very human unreadable, right? They're statistical measures of how noisy or spotty your images are. But with those measurements, if you do a dimensionality reduction, you suddenly, you'll see very neat clusters of cells. And if you gate them and look at and you back project them to your typical scatter plots, you go, oh, right, that's exactly the cells I was going to gate. But here they are, right? And I didn't need to go through that because they're just naturally separated out in this dimensionally reduced space. And we've even found that we don't need to do singlet gating, right? It just comes out from the spatial metrics. Here's the cluster of single cells, and here's the cluster of doubles and triples and so forth. So the workflow becomes still very human, but a lot more, the dynamic range is a lot higher. These clusters are extremely separate. Whereas if you try to gate them on a space of eccentricity against radial moment, you're still drawing a line through distributions, and naturally some cells fall one side of that or the other. Whereas if you work in a much higher dimensional space, you can avoid that.
Yeah, and still on the topic of analysis, we have a question from Stephen McClellan in the audience, who asked that given that in the past some systems such as the image stream didn't get widespread adoption because it was difficult to translate the kind of language of microscopy to cytometry experts, how is your software and your product dealing with this?
It's a very live problem. We're dealing with this by focusing on very clear applications. So right now we're focused on the application of manufacturing in T-cell therapies. So we're running very specific assays where we're looking at ratios of very specific cells. So we are presenting that data in simply the very typical format they're used to seeing it. Again, it's not about the image. I mean, if you don't need to even show people the image, we use the images and the data to derive the statistics on the populations of cells. So at the end of this, people should be able to call up scatter plots showing the total amount of that fluorescence against this fluorescence, color that by the cell type. And with the important difference that now that they can inspect those cells, click on them and get a very quick gallery pop up showing you that, yes, that is true. That's not an artifact. It wasn't something bizarre that's 2 cells stuck together in such a way that it did sneak through the singlet gate and had the same fluorescence. So you can visually inspect, but the software we've built so far has been quite well received by at least the 3 or 4 collaborators we're working with. Just through its simplicity, it's actually a very simple set of scatter plots and the ability to quickly pull up image data and, if you want to, create new parameters.
Well, I think maybe this is a good point to segue into your technology. And right, what do you think, Catherine? We should we've started talking about your technology, so let's hear more about it.
Great, and then you actually get to see what I'm talking about. For everyone here, this is not so much a presentation as a bunch of slides to help me talk through things. I guess that you could define that as a presentation. There we go. You can probably tell these come from my investor deck. Just single sentences and nice visuals. But quite, yes, this is a question you asked at the start about what is 3D imaging cytometry. And I've been leaning away from, although we specialize in 3D imaging cytometry, I do like to talk about it as spatial cytometry. And it really is trying to break that compromise between kind of classic microscopy and a classic cytometry. But there's no free lunch. You have to give up something. So it's not quite as fast as, the best analyzers. It's not quite as high resolution as the best microscopes. But you couldn't before get something that provided a reasonable 3-dimensional image of a cell at the scale of hundreds per second. So how do we do that? You're probably all wondering. So we do that using an optical technique called light field microscopy. It has other names: planoptic imaging, planoptic photography. You can even think of it as tomography. We take single cells, and when they pass through a laser that's interrogating it, causing fluorescence or scattering, we use a very high numerical aperture objective lens, the kind you would have in a high-end microscope, and we take the light that collects and we parcel that up into smaller lenses. So we have these micro lenses which chop that up and divert the light off into a nice, neat, straight line. Now, what those little lenses do is and it's quite simple, is that they capture the light that's coming from a very specific side angle from the lens. So these 7 pictures that we form are from extremely different angles. So the 2 images that are opposite each other, they are exactly 90 degrees apart. They're completely orthogonal. So you're seeing the cell from one side and the other. And the pair turned around next to it is also looking at them orthogonally. And the full angle of collection is something like 130 degrees, right? So it's extremely wide. And that's because we collect this view down the middle. So we have, if you still see my hands, we have these views that we pick orthogonally, and we have a view that we pick down the middle. And each view collects an angle of light. So the total amount of light you're collecting is a really wide angle. And that's what is really at the heart of all this. So we don't scan, there's no laser scanning, there's no laser modulation, that these lenses simply collect all the light, parcel it up, and redirect it to form nice, neat 7 images. And those images are just there on the sensor. And we use what's called a time-delayed integration sensor. It's the same approach used on the image stream, that this is a line scanner, so it's a line of pixels that ticks over at something like 400 kilohertz. But crucially, it's actually 128 of them or so. So the light collected is passed down to the next one and passed down to the next one, passed down to, so it adds up, right? So the whole time the cell's flowing over this chip, it's adding up the light that's emitting from it or being scattered. And that's where you get this beautiful sensitivity. You get this really high exposure. For a sensor that's running at 400 or 100 kilohertz, you're actually collecting a millisecond exposure. So the cell is underneath that laser light or LED light for a whole millisecond before it passes away. And there's no scanning. So for a whole millisecond, you're collecting that light. If you were to say, for example, use one of these other methods where you modulate the laser, you kind of change the frequency of the laser to computationally construct an image, you're getting an effective dwell time for each pixel more in the nanosecond regime, right? So you're going from milliseconds to nanoseconds. So the sensitivity difference is huge. Of course, the speed difference is also huge, right? So I mentally parcel up the imaging cytometry methods into computational ones and direct ones. So we are a direct one. The image stream is a direct one, and the FACS is a computational one. And I guess the ThinkSight compressive sensing one's computational as well. And these are all great techniques, and they're just approaching the field from different directions. Your applications will be a bit different depending on whether you want speed over sensitivity or spatial resolution over speed. So we have these 7 images, and we split them up, as you would do in any other system, into different spectral bands. We also have the scattered light and transmitted light. Our transmitted light's a little bit quirky because we have to have 7 transmitted light sources. You can't just put one light bulb, because then you would get one image. So we have a very bizarre looking bright field setup that produces 7 beautiful bright field images. And we then, or you can then, take that data. So this is what it looks like. I'll take a 3-color example where we're labeling the top green is some GFP bound to some cytosolic protein. The middle one is DAPI, and the bottom one is a condensate of interest, which is why it's got these little spots in it. And I should say this is the same cell. This is not 7 cells. This is the same cell seen from 7 different angles in 3 different colors. Now you can see where the data is piling up, right? So I've got, just for 3 colors, I've got 21 pictures. And down the bottom, I've got a different cell just to show you what the transmitted light looks like. And I used a doublet just to show you, you can see from a human point of view, you can see how interesting it is that you really do see around corners. This 3rd image from the left, it looks like a single cell simply because the second guy's hiding directly behind him. And maybe we don't have the right angle to see if they're actually touching or bound. I think they are in that second one. So we have this transmitted light data, but the system we've designed so far has 6 fluorescents and 2 label free, but we haven't poured concrete over that yet. We're discussing right now, how we should expand that, how many colors, what do the applications really need. The data from this is very manageable, with modern solid state hard drives, high compression. There's a lot of black pixels there, right? So you can losslessly compress this down quite well. So to give you a flavor of this, 150,000 cell experiment in 4 colors takes up like 10 gigabytes. So it's that some people that's big, some people that's small. I'll leave you to the audience to decide. You can fit it on a thumb drive, but you will have to wait a few minutes for it to download if it's in the cloud. And we can create the 3D volumes directly from that with a tomographic sort of deconvolution reconstruction. It's also why I hesitate to call ourselves fundamentally a 3D imaging cytometry, because at heart the raw data is multi-view, but I may be splitting mathematical hairs here that this is 3D in a different space. But the nice thing about the multi-view is it's very interpretable by modern 2D machine learned algorithms. So you can feed this into networks that have been trained on millions and millions and millions and billions of images and get out sort of sensible results. So it's very nice. We have these 2 spaces we can work in. We can work in the multiple 2D view space, where you can have parameters for each 2D view, or we can transform into the 3D space, which takes about a millisecond with GPUs. So we can turn it to 3D very quickly. If someone wants to write their own image analysis,
may I just interrupt with a question from
Yeah.
I mean, since you said you hesitate, to call it 3D imaging, but I mean, multi-view imaging. But I think I understood about your technology that actually the 3D reconstruction is pretty much unambiguous from that data. So it is a genuine 3D model that you are reconstructing.
Yes. Yeah. It's actually, light fields is fundamentally a 4-dimensional image. You capture X, Y, and what we call theta phi. So it's in the angle space. They come from 2 different angles. We happen to turn it into a straight line so that we can use a TDI line sensor. But we as humans don't think very well in X, Y, theta, phi space. So it's a mathematical transformation. But yes, you're right. It's all, I'd say I'm splitting hairs, but you are missing information. Like if you were doing tomography, if you've ever had the misfortune of being in an MRI scanner, it takes a long time because they take hundreds or thousands of angles, not just 7, right? So if you take 7 angles, your 3D volume is reconstructed fairly well, but obviously if you took more angles, it would be even better, more precisely. So these spots would be more precisely located if you took more angles. But if you take more angles, you split the light up more, and there's a sacrifice to sensitivity, there's a sacrifice to resolution. So this was our sweet spot. We did actually prototype a system that had 37 views. That was a nightmare to get working on the camera, but turns out, academically that's all very interesting. But 7 really hit the sweet spot for kind of practical, pragmatic system that's sensitive, fast, and gives reasonable resolution. There's a hand up from Patrick. Oh, sorry, was that Patrick's question?
I don't know. Well, I saw the hand up, but actually there's a QA. There's one in the QA as well.
Okay. So yes, sorry, I'm splitting hairs here. It is 3D. I guess what I'm maybe alluding to there is in our early days, we used the 3D all the time for everything. And now we use the multi-view more often than the 3D. We just find that easier to work with in terms of software and machine learning. It's a real thing. It's not just a schematic. We've actually built what's known as a technology readiness level 7 device. This is not production scale yet. This is a system that is good enough to take off site. So we put this in the back of a van, and we took this along to the Babraham Institute in Cambridge. We took it to the Stem Cell Institute, and we took it to GSK's site in Stevenage. And it spent months at these places just running through dozens and dozens and dozens of experiments. And we got to learn a lot about what people liked about the data, what they wanted to do with it, and we learned a lot about the engineering quirks and problems that we needed to fix in the fluid. And we've developed 2 software platforms, one for managing the collection of the data, acquiring it, and then one for post-acquisition analysis. So that top video is showing you a live experiment running where I think the event rate's ticking over at like varying between 80 and 150 events per second. And I think this is a quite straightforward T versus B 2-channel assay with a CD19, CD3. And at this point, in a matter of 4 or 5 minutes, it's up at the 25, 30,000 cells. And they're all beautifully in focus. We're using photoacoustic focusing, so it's very well focused compared to our early version. And the analysis software has gone through the ringer quite a few times, but at the heart of it remains scatter plots and gating. That's you're dealing with high parameter data. It's necessary in an imaging system to give people a portal to look at the cells and see the data. So this experiment here, I'm showing it's a cell cycle assay, and I'm just showing the dimensionality reduction there on not many parameters. We're talking like 12 spatial parameters that really pops out beautifully, these clusters, which if you click and inspect them, you'll see this is the mitotic cluster. The top cyan one are aggregates. The big orange blob are the GFP positive, and the big white blob are the GFP negative. And there was no need for any gating to do this. You just do the dimensionality reduction, and cells that look similar, land next to each other. But it's not a quantitative space. You can't take measurements here, so it's more of a tool to help people explore and annotate their data, and check that their gating is correct and they're happy with it. So we spent, I don't know, about 8 months and £1 million building this, which is pretty cheap I think, compared to American venture capital standards. And we're currently sort of raising more funds to be able to take this system and turn it more into an inline or at-line system for the cell therapy manufacturing industry.
Mhm.
What are we doing now? That's what I'm saying. So that was a segue into this, that we've pivoted a bit from being a general analyzer. Just being a company so small, it's hard to try to build a general product that will work well across all fluorescence and label-free assays. We found that what people really were blown away by on our demos is the label-free data, which is not something we set out to do. It just sort of pops out from the optics that when you collect these scatter images, if you have one scatter image, you get lots of interference and noise from the laser, a lot of speckle. If you collect multiple views and they're fused together into 3D, they make these beautiful, very high contrast structures where the laser's scattered off these points. Now, the biologists would ask me, what are these points? And I say, I haven't the faintest idea. It's not labeled. This is part of the problem, but it is giving a beautiful measurement of all the scattering centers in the cell for that specific laser. And so we found that, wow, we're getting great, really high contrast, clean 3D scatter data. And we started working with people to look at label-free phenotyping of T cells. And we're having fairly early success with that. So we put our results forward for an Innovate UK grant, and we were awarded £500,000 matched by Zeiss and Foresight. So £1 million is now going into developing the evidence that, not just developing the evidence, testing as well as developing evidence, the precision and accuracy to which we can replace cell therapy assays that currently use labels, with the idea being that you can simply run inline on a bioreactor, pull cells out and read out your CD4, CD8 ratios, pull out some measurement of potency or pull out some measurement of how many exhausted cells there are. So this is what we're doing now, which is a little bit thinner application space than all of our conversations so far. But it's just the nature of being a very small company.
Oh, I mean that's. I mean, of course, our final topic is about where it goes, and so and how what's the path what would it take to make this a major technique. But before we get to that, there are a few questions. I mean, I think, well, firstly, great presentation. There are a few questions in the Q&A. I think that just about the technique. And actually, I must say, although this is the investor presentation, it really brings it out, the sort of power as an analysis tool, because if you've got the clustering, and you can see those 3D images, they pop out a lot more than, say, images from a FACS Discover, for example. I mean, there's those the 3D revolving images, they're just so much richer. So, I mean, it's clear that, I mean, biologists would want to see, what's that cluster and it sort of leads to natural sort of hypotheses. What are we seeing there, and what's relevant in the image, and what can we interpret. So I think that's really that's really great, great demonstration. We should read out a couple of questions. Catherine, do you want to do it?
Go for one of them. So we have a question from Xin Yu about the applications that you've looked at. So it suggested that one interesting application could be distinguishing surface bound molecules from internalized ones, such as antibodies that could be on the surface or inside the cell. Is this something that you've already looked at or are planning to look at?
It was one of the very first things we looked at, and we were looking at GPCR internalization with some pharma companies. We were also looking at oligonucleotides, not just internalization, but then where do they end up, and where they degraded, how much of it's becoming bioavailable. Absolutely. We just found that space itself wasn't attractive enough or seen as a disruptive or big enough market for investors to fund the scaling of the technology. So that was absolutely where we went first, and we were very excited. But we have to go somewhere where we have a bigger impact. If we can crack the very, very difficult nuts of, label-free phenotyping, then that is a bigger opportunity. But you're bang on. That is where we spent, I'd say, the first year of our life exploring various internalization assays, because we found people that were very frustrated with data that they're trying to defend, whether something's genuinely been internalized or not.
Okay, thanks. I guess, Samson, you want me to go for the next one? We have, of course, comments about some excitement that CAR T-cell QC analysis is going to be really revolutionized by your technology. But the other question that we have is from PJ Chana, who's curious about the reproducibility of your label-free methods. So between instruments or between measurements, how much noise do you expect there to be versus the true kind of data?
We don't know yet. So we are running the first repeats of experiments like right now. What we have done is we've built a pipeline that's actually quite fast. I mean, training these types of models, I don't think we're going to have much success trying to create foundational models. I don't think they'll work in the tool space and the biology space, because we simply put, we won't get big enough, the question you're asking, repeatable enough data. I always try to remind people that get excited about AI that these large language models are using the entire sum of coherent human written language, which probably costs of the order of hundreds of trillions to generate. And none of it's noisy. No one's just mashing a keyboard, right? They're all words, they're all sentences. I'm sure some of those sentences are not high quality, but they're sentences. Data, well, you all know, like scientific data is some of it's crap, some of it's noisy, some of it's wrong. You've labeled the wrong thing, something went wrong in the protocol, and the scale of it's not big enough. So, you've touched a nerve here with your question. So I think the way forward is to build a pipeline that lets people build local models for their assay and their pipeline. Now, it has to be repeatable, but I think it has to be repeatable on their system during their month. That's what we have to find out working with people. It's like a calibration. Like you run your fluorescence assay, you have to do it in all your controls. Once a month, once a quarter, once every 6 months, once a year, and that's what we're trying to find out, is how stable this is. Now, of course, some people in our team hope that it's going to be super stable and we'll have a foundational model. It's so stable, we can just use it, and basically create a system that doesn't even have lasers, right? We can just sell people a much, much cheaper system that doesn't need to do all the fluorescence. And that's still possible. But we have a contingency plan, which is we're working out a workflow that allows people to fine tune. You call it fine tuning a foundational model, but basically build their own model, which means they have to have the fluorescence, they have to have the high-end GPU. So it does make the system more expensive, right? But it does mean that people will be able to have control over their own models and be able to tweak it as they like. If they change their assay, they can change their model.
Mm-hmm. Well, it's music to my ears, by the way, Kevin. I think let's not go down. Let's defend ourselves somewhat against these foundational models and stick to real science. The last topic, very importantly, what would this take to become a major technique in biology? Now, you mentioned you change your strategy and your investors and such like, but ISAC, we're largely interested in widespread techniques that are useful to biologists. So what are the challenges for this to be adopted widely? Is it technical? Is it the state of development, the maturity of your instrument and technology? Is it commercial? Is it about applications, demonstrating applications? how does it reflect, or say the product vision, the
Mhm.
features of the product, sorting and so on? How's that reflected in your strategy as well, for proceeding with the company?
Uh, so it might sound like a bit of a cop out, uh, but I wouldn't have left. I could have stayed in my role till I retired, running a lab and a cushy setting. But I believe that these tools are really inevitable, and if you could have that data, of course you're going to use it. And your question is, what would cause it to be mass adopted? It's like I think this is like a sort of pregnancy, and you can't really speed it up, right? But what things cause it to progress? Uh, it has to be slow. So I don't think it's good to jump in and go, here's a black box AI tool. So we always have to marry up the next step with the last one.
Well, actually, just to challenge you, I mean, say the technology that became the Fax Discover, that was snapped up by BD at very early
Hmm.
stage, of course, just based on what it could do and the conviction that that was
Hmm.
important. So, is it the case, say, that if it's technically mature, that it would be snapped up in the same way and pushed to be a widespread technique, or is it different somehow?
Well, in some ways, you're right. I think if I had limitless funds and could pursue for the next 4 years just making it robust and repeatable and scale it up and sell it, I have no doubt it would be taken up. Particularly because we're very much of an ethos of listening and designing something that people want and not just shoving through, this is high resolution, you're going to need it. Um, I don't really see anything, the barrier in its way other than the engineering required to make it turnkey, which I think would be about another 5 years, and maybe something of the order of 20 to 100 million pounds for a single company. But I think this is a bigger picture of entire community, which means all the companies doing this need to be kind of headed roughly in the same direction. So these are new types of assays. So what are the panels? And that's longer. That needs the early adopters to kind of triangulate and work together on how best to use these tools. So it's what you've seen in the past. Like, when did spectral first appear on the scene? These things take time, and I think it's going to iterate between technological barrier and then the application, not so much a barrier, but the application, triangulation, the protocols, ironing all that out. And it's a bit of a chicken and egg, right? So I think it's on us right now to make this work well enough that we can put it on the market for ideally everyone in this room to get one or apply for a grant to get one. And then we work together to what's next. Because I think that what's next is the convergence. My last slide was it's not about anything replacing anything. It's about enhancing the parameter space in which you can discriminate cells and discriminate measurements in assays. So I think it's about figuring that out together. And that's why it feels like a bit of a cop out saying that. But there's no major, there is no major barrier other than time and people working together.
Thanks, Kevin. Well, we're on the hour, so I think
Yeah, time.
that's all we have time for. Any burning follow-up questions, Catherine?
Not from my end.
Right, yeah. Well, really enjoyed it. Thank you very much,
No, thank you so
Kevin.
much for having me. And thank you
Yeah, no,
everybody for attending and taking time out your day to come, particularly if you're stuck in a hot room in France or Switzerland or in Oxford somewhere.
yeah. Well, so thanks everybody, and see you next time.